All right, so Assalamu alaikum and welcome back to the last subtopic of our chapter one, cells and its calculations. This is a highly important subtopic because you'll often see questions of calculations in your paper ones and in your paper twos. So this is a slightly technical topic and it can be difficult to understand for those who are a bit weak in maths. But just don't worry, we'll get through it, inshallah. And let's begin without any further ado. So, in the previous classes, we've already talked about the formula of magnification, that it is image size over actual size. We are now fair on the difference between image size and actual size. And I marked this portion as important because you'll be required to make lots of conversions while attempting the questions of magnification and the other topic which is now about to come. So if you have not learned these conversions, then please do so. So it can help you in attempting the questions. Okay, now moving towards the main topic, that is IP's gratitude and state micrometers. So the basic difference between stage micrometer and eyepiece gratitude. So basically, these are two scales. Okay. So the eyepiece gratitude scale. This is a scale which is present in your eyepiece. So eyepiece are the lens from which we view the specimen. We our eyes are kept at eyepiece lens, and then we are able to see the specimen which is placed on stage. So I have also sent the diagram of microscope on the WhatsApp group. So you can also see that where eyepiece lenses and where stages. So stage is the region where we put our slide with the proper specimen. Okay. So stage micrometer is the scale of stage, while eyepiece graticule is the scale of eyepiece lens. Now, we can skip this part right now because if we'll go into the depth of this English, then we'll be unable to understand the entire concept. So it's better that we practice this with our questions. And just one more thing that I would like to add here is that stage micrometer will always have a specific unit. It could be in millimeters, it could be in that is micrometers, but eyepiece graticule does not have any proper unit. The units of eyepiece graticule are arbitrary, so it does not have any units. Okay. Now moving towards this part, which is the question of your past paper. It says the diagram shows a stage micrometer viewed with an eyepiece graticule scale using a magnification of x400. So as per the previous slide, we can identify that this scale is stage micrometers, while this one with more numbers is eyepiece graticule. Okay, so we can also mark over here that this is stage micrometer and this one is eyepiece graticule. And okay. So they're asking that using the same magnification, a chloroplast is measured and found to be 4 eyepiece graticule divisions long. So they're saying that they measured chloroplast and it is 4 eyepiece graticule divisions long. So means somewhere here it is somewhere here it belongs. So they're asking how long exactly the chloroplast is. So basically they have given us the eyepiece graticule units. Now they're asking us the stage micrometer. They are now asking us the magnitude of stage micrometers. Okay, because we can see that the answers are given in proper units, which is So as far as this millimeters is concerned, we need to first convert this into Why? Because they have given us this which means that the answer that we are supposed to give, it should be in micrometers. So in order to convert millimeters to micrometers, this is going to be our first step, that we are converting the measurement of stage micrometers into It is multiplied by 1000. Okay, and you'll be unable to do this if you are not aware of this conversion. Okay. So in order to convert millimeters into we multiply the magnitude with with multiplication of 1000, and then we'll get our answer 100 So this is our conversion that we have established. Okay. So this is the this is the what do we say? This is the length of stage micrometers. That this is one division. One division of stage micrometers is equals to 100 UMs. Now we need to find out that how much is the magnitude of this unit of eyepiece gratitude. So we can see that this belongs to this line is ending at 90 and this line is beginning at 50. So we need to minus both of the values, which is 90 minus 50. In order to find out the difference between both of them, it is going to be 40. Okay. So this is 40 eyepiece gratitude units, while the stage micrometer that we have just calculated, it is 100 So this is going to be 40 eyepiece gratitude unit, EGU, because we do not have any solid unit for this. So this, with this being said, we are now able to establish our relationship, which is 100 of stage micrometers is equals to 40 eyepiece gratitude unit. 40 eyepiece gratitude unit. I'm just writing gratitude unit. Okay. Now, what they have asked us, we have established a relationship over here that 100 micrometers of stage and stage micrometer is equals to 40 eyepiece gratitude units. Now, they are asking us that using this entire magnification, we need to find out the stage micrometer value for chloroplast. So, we can now write 4 gratitude units. So this is something basically we do in maths, that we establish a relationship and then we are able to find out the fourth value using the methodology of cross multiplication. Okay. So this is exactly how we do in maths. So we are going to multiply 4 multiplied by 100. We are doing CM. CM means cross multiplication. 4 multiplied by 100 divided by 40. I'll also write the proper answer over here by using a calculator. I hope I'll be able to do it with this calculator. 4 multiplied by 100 is equals to 400 divided by 4 multiplied by 100 should be 400. Then I'm going to divide 400 with 40. So this is 10. 10 micrometers. Basically, I multiplied 4 with 100. It gave me an answer of 400. Then I divided 400 with 40. So my answer is 10. But once again, I'm stuck. They have not given me an answer in ordinary form. My answer is in ordinary form. This is 10 So I need to convert this value into scientific notation. And we all know that when we have a whole proper number without any decimal, then decimal is kept at, then decimal is placed towards the right side, and then we can move it. So our answer is going to be 1. Sorry, our answer is going to be 8. Okay, so this is how we are going to solve it. Now, moving towards other questions. So, it says the diagram shows a stage micrometer with divisions 0.1 millimeters apart. They have given us the same unit for a stage micrometer again. They're saying that stage micrometer has divisions of 0.1 millimeters apart, which means we again convert it into Why? Because they have given us something in so we are going to convert it. So it's a basic rule that you never calibrate values when they are in different units. You need to make the units same in order to calibrate any value. This applies for maths, this applies for biology. So this is a common maths rule. Okay, so you're going to multiply 0.1 with 1000 again to get us an answer of It is going to be 100 like we did in previous part. So now this is 100 Okay. So we'll do the same thing again. Since this is stage micrometer, we need to find out the value for eyepiece gratitude as well. That 100 is equals to how many eyepiece gratitude units. So we'll do the same thing again. We have the same calculation over here as well, that it's 90 and this is 50. So in order to find the difference between this division, we are going to subtract 90 minus 50. And our answer will be 40. So again, the same relationship that we established in the previous part. We are going to establish the same relation over here as well. So this is just by chance that I got questions with same calibration relationship. This is not going to happen in all the questions. You will get different conversions as well. You will get different values as well. So this is just by luck. So this is now 40 reticule units. ठीक है? So 90 minus 50 is 40. Now they're asking something different. In the previous part, they gave us the value for gratitude units and we're asking us the value for stage micrometers. Now they're saying the same eyepiece is now used to examine a blood smear. How many gratitude divisions will cover the diameter of a white cell of 10 Since we have a solid unit over here, we can identify that this is a stage micrometers value and they are asking us about the gratitude divisions. So they are asking us the arbitrary value and they have already given us the stage micrometers value, which is 10 So I'm now going to place 10 under 100 because I need to find out the fourth value, which should be in gratitude units. So now I'm going to establish the same relationship. I'm going to perform the same steps. I'm going to cross multiply it. So over here, I'm going to multiply it with 10 multiplied by 40 and then divide it by 100. So we'll do it again. Just give me a minute. 10 multiplied by 40. 10 multiplied by 40. It's 400. And then we'll divide 400. We'll divide 400 with 100. And our value is 4. Okay, so this is our answer. I multiply 10 multiplied by 40. It's 400. Then 400 divided by 100. So our answer is 4. Okay, so this is it. So we need to, in order to grab this topic in a better manner, we need to practice a lot. Okay, but however, I have covered the entire concept. We're going to do the same thing again in all the questions that you'll get. Okay, so do not get confused. I am going to solve one more past paper question with you guys, and then we'll call it a day. Also, we already did this one. I already did this question with you guys. Now, let's see. All right. So, this is not the same question, but I wanted to tell something about it. That the diagram shows a high power drawing of a plant cell. The actual length of the cell between X and Y was 160 So they have given you the actual length, okay, between X and Y, which is 160 Now they're asking you the magnification of the cell. So we all know that the formula of magnification is image size over actual size. Okay? So they have already given you actual size. You do not need to worry about it. But what you need to worry about is your image size. So how are we going to calculate image size? It is by placing your scale between X. You'll place your scale here, in here, and then you'll measure the length. So this is going to be your image size. Okay. And the method and the way that you're going to put your scale is that this region should have 0 centimeters pointing towards this side, and whatever, let's say you get 5 cm or 6 cm. Not sure what value will you get, but this is how you're going to do when your image size is missing. You need to place your scale and then you'll be able to find out image size. And then you'll keep image size over here, actual size over here. And one more thing, you'll have to convert the value because your scale gives value in cm. Okay, so be mindful about these conversions. These conversions are highly important. It's high time that you start revising these conversions so that you could remember it. Okay, so your scale is going to give you value in centimeters and you'll convert it into Only then you'll be able to find out the right answer. If you put your centimeter value over here and you put your actual value over here, which is in then you'll get a completely wrong answer. So be mindful when you are handling the questions of magnification. Surely you'll get conversions in these questions. Now let's move on to the other question. All right, so we'll do this question and then we'll call it a day. So this is saying the diagram shows a stage micrometer on which the small divisions are 0.1 millimeters. Again, the same thing, stage micrometer. And again, this division is of 0.1 millimeters. 0.1 millimeters. It is viewed through an eyepiece graticule. It is viewed through an eyepiece containing a graticule. Now they're saying the stage micrometer, and this is replaced by a slide of a plant cell. So first, initially, we need to establish a relationship, which is that, so we can see that answers are given in so we'll convert it again, multiply by 1,000, we'll multiply by 1,000, and we'll get a converted value of 100 100 UM. And then again, we'll find out how many IPs graticule is this. So this is again the same conversion, I believe 90 minus 50. It is going to be 40 graticule units. So again, the same relationship. Again, this is by chance. So we can say that 100 UM is equals to 40 graticule units. But now there is some confusion that now we cannot just directly put our value, our third value under this relation and find out the actual value. They have given us a slightly technical question, which is that they are saying that stage micrometer is replaced, that they have replaced this with a slide of a plant cell, and now they're asking us the width of chloroplast. So this is a technical question. Achha. So see, this is a relationship, but this is not a definite relationship. When we're finding out definite relationships of maths, we need to establish those with the unit of 1. Now we are going to find out the value of stage micrometer with respect to 1 graticule unit. कि एक graticule unit कितना stage micrometers occupy करता है. I'll explain you the reason. You'll come to know about it. So we are going to cross multiply it again. 1 multiplied by 100, 100, and 100 divided by 40. So let me do it. 100 divided by 40. This is 2.5. So you are getting a value of 2.5 of stage micrometers. So 1 graticule is occupying 2.5 micrometer divisions of stage micrometer. Now they are asking you, so now this is your solid relationship. Okay, this is your defined relationship. A graticule unit ने कितना occupy करा. Now they are asking, because they have replaced the stage micrometer with the slide of plant cell. So what you are going to do is that you need to find out the width of a chloroplast. So if we examine this plant cell, we can see multiple chloroplasts over here. Yeah, but the most we'll try to identify the one with proper values. So this is a chloroplast, this is also a chloroplast, this is also a chloroplast, but this is not a chloroplast, it's a nucleus because it's slightly larger. Okay. So I'll try to draw a line, but I'm not sure if it is going to be accurate or not. Let's see. Okay. This is one. Now let's see how many units of IV's graticule this chloroplast is occupying. So I can see that 1, 2, 3, 4. This is occupying 4 eyepiece graticule units. So I'll write 4 over here. This is under 1. I'll write 4 graticule units. If I'll do it for some other chloroplast, I'll get the same value. Okay, so you won't get a difference in values. So let's do it. The calculator. I am going to multiply 4, multiply by 2.5. I get a value of 10, and then 10 divided by 1, which is going to be 10, obviously. So our answer is 10 UM. All right. So this is how we are going to solve the questions of IPs, graticules, and stage micrometers. And I hope you're able to understand the concept because we'll practice more during our yearly sessions of past papers. And with this being said, we'll also continue past papers of cell again tomorrow, and then we'll begin with the chapter of cell membrane, inshallah. Okay. So I'm calling it a day over here. Take care and Jazakallah khair for joining.